So, good morning, everybody, professors. I am Shiro Futaki from Kyoto University. And first of all, I'd like to thank to all of you for selecting me or electing me as a honorary member of Hungarian Academy of Sciences. It's my very great honor and very happy to be here. And also, I had a chance to make a collaboration with Professor Fudes. So it's my, I guess, 5-6 times on the Budapest. It's always very beautiful city, so I'm very also happy to come here again. So I'd like to say, and also Professor Philip, thank you very much for your kind introduction. And today, I'd like to talk about my peptide-based approaches for delivering exogenous molecule into cells. And before going into that particular topics, I'd like to introduce just my brief history briefly. I was educated as a synthetic peptide chemist in the Kyoto University, and the mentor was Professor Yajima. At that time, solid-phase peptide synthesis is not popular in Japan, so we had to make in the solution phase. So that kind of laborious, I should not say, but so we did kind of step-by-step synthesis, and finally they make some fragment of peptide, and then conjugation of them get a full-sized peptide. But in one sense, I was very lucky because at that time, to make this 30 amino acid residues, I needed 3 years, and just I got PhD. Nowadays, you know, the solid-phase peptide synthesis become very popular. You can synthesize with this peptide only for a week. So you have to worry about many other things to get PhD. But anyway, and when I go to the Tokushima is small town, a city, I can say, in Japan, that is Shikoku Island. You know, the Japan is composed of 4 major islands, and Shikoku is the smallest, big island. And that is 2 hours from Osaka, like that. And I spent there 10 years. And at that time, solid-phase peptide synthesis becomes more popular, especially FMOC synthesis. So I was able to make synthesis peptide more easily than before. So what I want is to prepare functional peptide. But as you know, peptide itself doesn't take a specific conformation. So you need some devices or some trick to allow peptide to form a special conformation to make a function, exert their functions. So in such case, I'm interested in making a kind of ion channel peptide. So membrane can be a support of peptide and make them the kind of helical structure and made a pore in the membrane. And then after staying 10 years in Tokushima, I came back to Kyoto. Kyoto is also here in the almost center of Japan. And wanted to be more biology-oriented project. And today's talk is like to introduce my research in when I came back to Kyoto, after I came back to Tokyo. So first topic is membrane penetration of arginine-rich peptides and lipid packing defect. And second topic is attenuated cationic amphiphilic lytic peptides, so it's long terminology, as a unique platform for intracellular delivery. So when I started my new project, that is to deliver biofunctional peptide or protein to control cellular function. Saying that is very easy, but in fact, peptide and protein are very difficult to deliver into cells efficiently. I used to various liposome delivering agent, but that was unsuccessful. One of my friends taught me that the peptide derived from HIV So human immunodeficient virus 1, TAT peptide, can have ability to bring the exogenous protein peptide into cell very efficiently. The sequence is here. So sorry, but it is written in the one letter code, so maybe some people are not familiar to writing this amino acid, but what is important, R is arginine, and K is lysine. So that is, the arginine has a guanidine function in the side chain, and K has a lysine in the side chain. So this has about 10 residue peptide, central part of this sequence, about 10 amino acid residue, amino acid, and that the, say, more than, we have 6 arginine and 2 lysines in the sequence. It's cationic and very hydrophilic. So in my idea, membrane is kind of a hydrophobic barrier to protect the cell against, say, to protect inside and outside of the cell. But this is very hydrophilic and basic, very really soluble. So first, I doubt the, say, reality, or say, possibility of this ability of this peptide to go through the membrane. But at that time, there are many reports, several reports that show the success of the using peptide. So I mean, making a conjugate of TAT and other small peptide, a protein. They just make a conjugation and apply them to the culture media. They eventually lead the cell as a functional control of the cells. That was looks very fantastic. Yeah, arginine is here, like here, and lysine is here. It's too basic. So I first check if this story is really correct or not. So what as I introduced, I'm a synthetic peptide chemist, so synthetic peptide is fun for me. And maybe not very fun for my student, but this is the original TAT peptide, yes. And the HIV-derived peptide sounds kind of mysterious, so maybe first my idea is there are some specific receptors or specific mechanism to allow them to get into the cell. So I'm get a PhD in the pharmacy. So what we can do is just form a D form of the TAT peptide. If they are the receptor, there's a chirality. So D form of TAT must not work. But actually, this was ability go into the cells. And also, I made all the amino acid here to change all the amino acid here to arginine. And this also has a function to get into the cells. Well, it's funny. So I wanted to say, find a kind of exception or rule that cannot go into cells. So my laboratory also at that time involved in the design of RNA or DNA binding peptide. And the TAT peptide is also originally from the RNA binding area of a segment of HIV. So I made various RNA binding peptide. And eventually, I made 10 peptide and 9 peptides except one. This was able to get into the cells. And if I see, but they got into the cell, but as say, we check the efficacy. The ones that contain more arginine has a better translocation. So eventually, I found that the arginine should play an important role for this kind of peptide internalization. And after that, I had more interest in the studying the mechanism rather than application aspect of the peptide. So for example, I made kind of branched chain peptide, and they also get in. And that means not only the arginine, but also linear structure are not important, but the kind of cluster of arginine should be important. So eventually, our work, or other persons' work stimulated many people, and many of the kind of various type of peptides, including beta peptide or peptoid or carbamate, shown to have ability. So they have they commonly have the kind of guanidine function in molecules. And the similar idea independently proposed by Professor Bender, Paul Bender in Stanford University. And he is kind to make a kind of review and the account of chemical research in the American Chemical Society. He suggested me, he picked up the 3 major or most popular cell penetration peptide paper. That one is mine. So this is the 2013 paper. And nowadays, I'm happy to have a paper, this first paper cited more than 1,000 times. So, as we studied rather seriously about the mechanism of translocation, and eventually we found that this is arginine peptide or tartott peptide and connected with your biofunctional molecule. And if you choose the conditions, they can go directly through plasma membrane, as I introduced. And also, in the most cases, they can taken up into cells by so to say, endocytosis. That is physiological uptake method of the cell to get nutrition or kind of detoxification of other, for example, bacteria or something. Then today, so I mainly focus on this mechanism of direct penetration through plasma membrane. Okay, so you can say that this is a physiological method of uptake, and this can be kind of more physicochemical uptake method. And if you understand, if you go into the arginine peptide, the cargo can go, if they go through the membrane, direct through the membrane, immediately they can spread into the cytoplasm side all over the cellular compartment. That is very efficient. And this is a dose response relationship of peptide, arginine peptide. So we use fluorescent labeled arginine 12, this type peptide in this case. And the vertical line is the cellular uptake amount. When we increase the concentration of peptide, and then it's cellular uptake grows up very slow, growing up very slowly. But when we get some kind of threshold, concentration, it steeply, cellular uptake steeply goes up. And the detailed study elucidated that this area, cell is mainly, peptides mainly taken up into the physiological endocytosis method. But once you exceed the certain concentration, maybe probably the ability of endocytosis is saturated. So peptide cannot get more taken up into cell by that method, and then accumulation peptide on cell surface allow the peptide to influx into the cell by going through plasma membrane or cell membrane. So you see this kind of spread signal immediately. But you say that polyarginine is kind of detergent. So you can think that this is just simply peptide perturbing the plasma membrane. That this is not true. When we try to say the intactness of plasma membrane by several methods, plasma membrane is also okay. So we have some, we conclude that we have some special mechanism that allow peptide to go into the cell through plasma membrane. And a friend of mine who is Stefan Mattel, he is a professor of Geneva, University of Geneva, suggest me the kind of interesting molecule. That is pyrene butyrate. This has a hydrophobic moiety with carboxylate. When we pre-treat the cell with this compound, we have a very fantastic method of internalization peptide. So to treat the cell with this agent and add arginine peptide, we immediately see the influx of peptide into cells, and 5 minutes later, all of the cells are the signal of peptide, like this, very quick. And if we are lucky in some cases, so this is in the case of GFP conjugate to polyarginine, in the presence of pyrene butyrate for this conjugate, fusion protein come into the neural cells. And you know, these cells are very difficult to get to gene transfection. That means our work using this system, the highly efficacy delivery was attained. Our original idea or understanding of how this molecule proceed as a promote the cellular uptake of this polyarginine like that. Inside of the cell is negatively voltages, you know, and this is cationic peptide. So like electric current going to high voltage plate to the low voltage plate. But you know, the cell membrane is a hydrophobic barrier to prevent this cationic guy go into the cells. But if this kind of hydrophobic moiety with cationic charges were waiting this guy to come into the cell, maybe this hydrocarboxylate can form the kind of hydrophobic, can form a ion pair with this guanide function of arginine, and they make a kind of a complex, transient complex. So if this molecule of course has a kind of a hydrogen bonding or ion pair with water or solute molecules, but if this peptide go directly into the cells, they have to decrease that kind of energy favorability of the ion pair or hydrogen bond. But if this molecule are already in the cells, they can switch this kind of ion pair or hydrogen bond to this. So that should be decrease the energy barrier of this peptide goes through the membrane. That is our one possible explanation, and I think it is partially true. It is also correct. But we also have another idea of this to promote translocation. We found that the 18 amino acid related amphiphilic peptide derived from the kind of endocytose promoting peptide has the ability to induce positive curvature. That means in the membrane. That means membrane, this hemophilic peptide inside the cell membrane, and these make the kind of steric hindrance to the surrounding lipid molecule. So this push these peptide, push these lipid molecule away from this peptide. That makes the surface area on the extracellular leaflet becomes larger than inside of the cell. That eventually peptide, membrane has a kind of positive curvature, or the positive curvature with the interaction with this peptide. So what happened when the positive curvature is induced in the membrane? So you know the membrane usually has a kind of packed structure, but sometimes acyl group is exposed because the acyl group is thermodynamically moving. So there is kind of a defect, so to say, patching defect that lead the acyl group exposed onto the cell surface. But if you make the curvature, and assuming if the same numbers of lipid are composed on the same molecule, say in the lipid membrane, the curvature should be larger. Area or number of the defect becomes increased. And the polyarginine is said to make a kind of a hydrogen bond with this lipid head. So this is the lipid head or lipid phosphate in the membrane. Arginine guanide functions here. So this has a very suitable hydrogen bond and also ion pairs. That is said to be the one explanation that polyarginine efficiently goes through the membrane. But even if this hydrogen bond is ion pair is formed, arginine peptide can interact lipid membrane. That is just peptide is concentrated, adsorbed on cell surface, and that doesn't tell you how, say, the peptide translocation is promoted. And if you see the signal as a structure of polyarginine like this, guanide function, and of course, this area is hydrophilic. But if you see this part very well, trimethylene, that is comparable to valine, and the main chain. That means although this part is hydrophilic, this part is rather less hydrophilic or rather hydrophobic. So if the guanidine function make an interaction with phosphate and they recruited the cell surface, and the more curvature leads the more exposure of the membrane defect or acyl chain on water air surface or membrane surface, this area can have more chance to interact with lipid core. So if we say as long as this peptide goes through the membrane, lipid core have to go also. Peptide chain, main chain have to go through the lipid core. So if the acyl exposure of the acyl chain more facilitated the interaction of hydrophobic part of the peptide, then that will also promote the translocation of the peptide. That is my idea. But it is to show, understand this is not a simple case. We did kind of analysis that using surrounding sensitive dye named di4-anep DHQ. This has a kind of this structure, and also fluorescence is shifted depending on the condition. If the dye is placed in the hydrophobic condition, they have a shift in the 500 something. But if this peptide is placed in a more hydrophilic condition surrounding, this has a rate shift to make a maximum and 600 or something. So if the lipid has a lot of defect, more defect, this should be, acyl chain has more chance to expose to water. That means the lipid core becomes more hydrophilic. So we should see some kind of rate shift. And we can analyze the extent of the exposure in terms of the, so to say, GP value analysis. And this is in the case of the cell treated with the epsin peptide. That is what I said before, that is curvature-inducing peptide. And SAR1P is also the endosome-related peptide, and that also has the ability to induce positive curvature. We set several peptides and also find that these two peptides having, say, this peptide has the ability to facilitate polyarginine into cells, also has the ability to lose the packing state, lipid packing. Also, I introduced you the pyrene butyrate as a hydrophobic counter-anion. But we also realized that peptide also have ability to make a induce curvature or tubular structure in the cells. And the physicochemical study using DSC, dynamic differential scanning calorimetry, said that the peptide also has the ability to positively to induce positive curvature in the cells. So when we treat the cells with pyrene butyrate, there is a very big decrease of the GP value. That means lipid packing is very decreased in the presence of pyrene butyrate. And then also we saw the kind of efficient influx of polyarginine. I can say that probably or presumably because this carboxylate is negatively charged carboxylate stays on the cell surfaces. But this is inside of the lipid core. But this has a kind of rotation. Then this has a kind of certain steric hindrance. So this guy is rotated in the on the cellular surfaces. This makes the more lipid packing defect. That we thought. And if we are checking if this peptide this molecule really make a curvature in the actual cells, we use the kind of devices named analytical method uses scanning ion conductance spectral microscopy. You probably know the AFM, atomic force microscopy, that use the kind of tipping needle, say, studying on the searching on cell surface. But this peptide, this microscope doesn't use the actual needle, but it uses the kind of electrode. And they have some kind of, say, electrolyte coming through this pipette, glass pipette. And if the distance between the cell and the pipette is constant, we'll get a constant, similar conductance in this influx. So we can, without touching directly to the cell surface, we can analyze the distance from the pipette from cell surfaces. That eventually we can understand the cellular structure or height of the with this using this peptide, this machinery. And this is a little bit difficult to see, but sorry. But so when we treat the cells with the pyrene butyrate, we see it's kind of, without doing that, we don't see very big changes. But when we treat the cell with this pyrene butyrate, we see some kind of difference. For example, you need the kind of red colors, arrow. That is the place that increase the height, and also green place that decrease the height. That means the the membrane surface becomes up and down due to the in the presence of this pyrene butyrate treatment. And this is more easier to see using this kind of line scanning, and we actually see the curvature was a kind of waving structure is formed in the cells by using the pyrene butyrate. So in a summary, as summary of first talk, I think that the international mechanism of arginine-rich cell penetration peptide includes direct cell penetration, membrane penetration, and also endocytosis. And the membrane curvature induction and lipid packing loosening may facilitate the membrane translocation of arginine cell penetration peptide. So I'm switched to the second short talk and subtopics. So arginine peptide has the ability, as I said, maybe as a part of the plasma, as a little bit ability to translocate through the plasma membrane. And if you attach relatively small molecule with this, they can go also through the membrane. That is good. But whenever I make a talk, such a talk in Japan, pharmaceutical company people always ask me one question. If the conjugation of polyarginine with antibody allow the membrane translocation antibody, I'm a very honestly person and say no, because in this case, polyarginine make a kind of perturbation or structural alternation membrane very locally. And if the molecule attached to this is relatively small, this can go through the membrane, while the membrane structure is alternating or changing by this, affecting by the polyarginine. But in this case, antibody's molecular mass is about 150 kilodalton. That is very big. So even though this molecule part of the membrane was or change the membrane structure, local structure, this is too big to go through the membrane. So I'm always says, oh, I'm sorry, no. But one day I become kind of offended because why? This is not my fault. This is peptide fault, but why I have to keep saying sorry to the pharmaceutical company people? Then I think I should need some other method to allow the antibody get into the cell more efficiently. That is the second talk. And if you deliver protein into cell, like CPP is one choice. But as I said, if the molecule is so big like antibody, this is not realistic because it is difficult. And if you force them to go through this membrane, you need certain size of pore, defect of membrane that allows this big molecule go into the cells. That also, if this large protein gets into the cell, smaller protein inside the cells also gets out. That is very toxic to cell. So this idea, direct say, membrane penetration, like a physicochemical interaction, may not be suitable for the antibody delivery. Alternatively, people use, so to say, endocytosis as a route to the delivery of macromolecules. So gene delivery is also using this pathway. So in this case, with that part, we say maybe if you molecule of interest is trapped in this, so to say, endosome, then if you have some idea to disturb the endosomal membrane, this leads you the escape of this kind of molecule of interest. But in this case, you need kind of selective perturbation method of endosomal membrane over plasma membrane. If there's no selection, the agent, say, delivery agent also part of the endosomal membrane that leads the same, say, toxic effects to cells. So to obtain this kind of selection, what people used most is the difference in pH. So the inside of the endosome is known to be acidic due to the protons channel is appearing on the endosomal membrane, and outside cells is neutral. So there are so many, so to say, pH-sensitive peptide protein or polymers have been developed. And I think these are good to use for the delivery for gene or something, because only small, tiny amount, liberation of tiny amount of nucleic acid in the cytoplasm, they often get their satisfactory result. We often get a satisfactory result. But in the case of protein, protein cannot have ability to recycle in the cell, so we need to say much more efficient method to allow this endocytosed protein get into the cells. And when it comes to the ability to perturb the plasma membrane, this is the, so to say, cationic amphiphilic peptide or antimicrobial peptide has a very powerful power. They make a kind of aggregate or assembly structure on cell surfaces of cell, and then make a pore on the cell, and then kill the cell, for example, bacterial cells. But this is too toxic for the other cells. Often they have kind of cell lysate ability to electrolyte, right, or something. So people say less often use them for drug delivery. So I thought the mechanism of this kind of cationic peptide to pass out the membrane is the kind of hydrophobic, so peptide to form helical structure, and the hydrophobic face of the peptide interact with the plasma membrane and make a assembly and leading to make a pore or defect or rupture formation on the cells. So if we introduce some negatively charged, for example, glutamate on the potential hydrophobic site of the peptide. Maybe this, if we are lucky, this decreases the hydrophobic interaction of the peptide to plasma membrane cell surfaces. But if we keep that net charges of peptide positive, and you know the cell surface is rather negatively charged, the peptide will adsorb on the cell surface, and while making, not making the severe perturbation on cell surface, they are taken up in endosome. And at the endosomal membrane, if the acidic membrane will make the protonation of glutamate to glutamic acid, then hydrophobicity will increase and the peptide recover its original perturbation ability and allow the release of the your molecule of interest. That is, if we get that kind of story, we should be very happy. But this is a very simple idea, and I thought many people have already tried it. But as long as I check the literature, almost nobody, or actually nobody, tried to that style. Because maybe people thought that this lytic peptide, the ability of cell lysis is very strong, so it's not good for the drug delivery or protein delivery. So after selection of several, so to say, lytic peptide, okay, we found that the M-lycotoxin, the 25 amino acid residue peptide, should be one of the most toxic to cell. This has peptides, blue one, lysine, is like a cationic peptide, amino acid, and yellow one, leucine, phenylalanine, tryptophan, hydrophobic amino acid. So this peptide doesn't have very typical or very clear amphiphilic structure, but this also has so-so amphiphilic structure. So this is a cartoon. So when we make an analog of this peptide, and this has that by inserting or substituting the glutamic acid in the potential site of the helices, like this. Now we made one substitution mutant and two substitution mutant of glutamic acid. And by the way, what people asked me about this talk is this: what is the origin of this M-lycotoxin? This is a kind of spider toxin, and the name of California. But so I'm happy. I sorted this sequence by internet search, so I don't have to see this ugly creature. But I also don't like to see, of course, this kind of creature too. Okay. So, but we simply say insertion of just one glutamic acid. But the result was very fantastic. There are clear mapped difference between original one and the substituted mutant. So this original peptide has a cellular toxicity about EC50 is 1.3 micromolar. This means this, say if you treat the cell with 1.3 micromolar peptide, 50% of cell is killed by cell lysis. But the insertion of the one peptide decrease, there's a significant decrease in cytotoxicity. And just one insertion make the EC50 more than 40. That means singular glutamic acid insertion in the potential hydrophobic site reduce the cytotoxicity on 20-30-fold. And I skipped all the intermediate study, but eventually this peptide works for the antibody delivery. So we found that this is a red-colored protein labeled intracellular model protein, and this is protein delivered from antibody delivered outside of the cell. So if this is a confocal, so to say, confocal microscopic analysis. So if red-colored protein is co-localized with green color, we should have yellow color. Green plus red becomes yellow. So we see happy to see yellow color in here. But without saying, when we deliver the other enzyme, other antibodies, non-specific antibody, we don't see this kind of yellow color significantly. And also, we pre-treat the cells with the glucocorticoid receptor antibody. And if we treat the cell with dexamethasone, that is a steroid. That is, treatment of dexamethasone leads the expression of some several genes, and the glucocorticoid receptor works for the activation of other intracellular receptors, intranuclear receptors. And the pre-treatment of the cells with anti-glucocorticoid receptor significantly reduce the activation of this gene expression. That maybe that suggests that the possibility that our story is usable, applicable to the control of cell biological signal using a specific protein to target in cellular antibody. But when we do analyze the mechanism of L17, we use a kind of different or new finding. So when this is what we see, the actin structure, cytoskeletal protein structure in the cell. This is live cells. And without L17E, we don't see, we have some change, but we don't see a very specific, very big change. But when we treat the cells with L17, please see the yellow circle there. You see some kind of veil-like structure of the with forming around this area. That means, so to say, this suggests that the actin polymerization push the membrane outside to the cell, and this is, so to say, lamellipodia formation. That makes the treatment of cell with this peptide really make a structural alternation membrane structure very much. And also when we try incubate the cell, and we do the kind of time course analysis, and when we treat the cells with the, for example, dextran, this is a model of biopolymers, in the 5 minutes in the presence of L17E, and wash everything out, and then keep watching the cells. And this is 5 minutes, this is 15 minutes, this is 30 minutes, this is 16 minutes. And if dextran is taken up in endosome, and then released along with endosomal maturation, so to say endosomal maturation, we need maybe 10, 15, 30 minutes to allow the peptide go, dextran get out from the endosome. But that means we have to wait endosomal maturation. But the result was not that like. So this is the percentage of cells having the diffused dextran signal throughout the cell. This is this treatment, you already get 40% of release of dextran to 40% of cells. But even the prolonged incubation doesn't yield the increase of the cellular diffuse signals, cells with diffuse signal. And we also need to understand this is physiological machinery is important. In the 4-degree ATP is not produced and the endocytosis is not operated. Or in the case of APP deficient conditions, we don't see any. Under these conditions, we don't see any internalized the kind of dextran signals. So something energy dependent, but not very different from the typical endocytosis inducing by our peptide. That is what we thought. So we more precisely analyze the method than dextran translocation into cells. And when it comes to actin, as you show me that the actin, when actin is polymerized, there should be, we have some inhibitor to prevent that. So this is cytokeratin, and also EIPA is a inhibitor to prevent the actin polymerization and so the membrane wrapped structure of the membrane. And Wortmann is another inhibitor to prevent the fusion of wrapped membrane through the membrane. So you see the kind of movement of membrane like that stage, and that eventually lead the fusion of cell membrane to encapsulate big extracellular material into cells, and that is so to say macropinosome. And when we treat the cells with these inhibitors, we have a significant decrease of this cellular diffuse of the dextran. That means actin polymerization is needed to this promoted dextran translocation. But in this case, when we treat the cells with Wortmann, we don't see any decrease in the cellular uptake. That means, so this is a membrane fusion to form the actual vesicle, endosomal vesicle may not be needed, but the wrapped structure themselves are important to delivery to translocation of the dextran polymers. So what we, in conclusion, we thought that the peptide lead kind of membrane wrapped structure in the membrane, and the membrane structure is always changing in this wrapping structure, and packing also becomes looser. And in such case, peptide may interact with membrane, maybe transiently make a cleft defect or rupture in this wrapped membrane. But while the rupture is stopped, we don't see any internalization or influx of peptide. So this concept is maybe different from the traditional peptide membrane interaction, so to say antimicrobial peptide. That is the kind of, we can analyze in terms of physical-chemical mechanism of the internalization, physical interaction for analyzing the perturbation of the amphiphilic antimicrobial peptide membrane perturbation. And this is a typical, and also endocytosomal escape is one of the typical approach in the drug delivery person. But this is needs maturation of endosome. So this doesn't meet. So maybe we found a kind of new aspects of delivery mechanism that may be applicable to the large molecules such as antibody. Similarly, with this, again, this is similar cartoon. When membrane curvature is induced by actin, not by, say, curvature-inducing peptide, despite the actin. So the actin pushes the membrane upside, so you have a larger defect or looser liquid packing on cell side. And the peptides can interact, so amphiphilic peptide, cationic amphiphilic L7T can interact with this membrane. Then this makes a transient perturbation on this wrapped membrane. Without this have a very probably has a less activity, this guy. So the defect of packing, say, loosening is needed to interact to the L7TE. And then this transient perturbation allows the influx of the antibody other macromolecule into cells. And we also analyze the possibility of that using the same trick, and we see the significant decrease of the membrane packing when we treat the cell with L17E. That means our peptide has the ability to make the repeat packing loosening and also lead the transient perturbation of cell membrane. So this should be, I hope, the kind of new idea of the that may be applicable to drug delivery. And we also make advanced new types or advanced improved type of peptide delta HAAD. This has a kind of two substitution, histidine to alanine, and also glutamate to amino-arginic acid. So due to the time limitation, I all skip the national array, but eventually I can say this peptide has a higher helical structure and also higher pH dependence. So this peptide not only perturb the not only yield a similar effect to L70 on cell surfaces against ruffled membrane, and also this peptide has the ability to perturb the endosomal membrane as well. So eventually, this ability of this peptide, delivery ability of new peptides is higher. So we see the 75% of the release of the dextran labeled cells compared with, that is far better than the original L70E. And also this is time course study, and since the L70 doesn't make a significant increase in the when time passed, but this peptide has a kind of increased ability to cytosolic release of the dextran depending on time. That means the maturation and endosome is needed or helps the peptide works to say deliver the dextran or other molecule into cytoplasm. And that means we can get the more interaction, more bioactivity than this peptide. So finally, so this is the summary. So mechanism of action of L70 includes transient membrane permeation through the ruffling membrane. This is the energy-depending pathway. And enhancing hydrophobicity and pH sensitivity yield more efficient delivery tool, HAL. So sorry, this time I skipped most of this part. And attenuated cationic peptide, I mean the insertion of glutamate or other negatively charged molecule to the amphiphilic lytic peptide may provide a unique platform to intracellular delivery. And this is almost my end of my talk, but before that, yeah, I can explain the Kyoto. Yesterday I made a similar slide, and the people know the name of Kyoto. Kyoto, in the Chinese character, we like this. And Kyo means this first Chinese character, To means second character. And both the Kyo means capital, and To means also capital. So Kyoto people, I'm a Kyoto person and very proud of the Kyoto is the capital of capital. Maybe you know Tokyo better than Kyoto, but Tokyo means To is, we say, east, and Kyo is the capital. That is just east capital. So the capital of capital, yeah, this is actually Kyoto had been the capital of Japan over 1,000 years. This is just 100 years and something. So that's how Kyoto people like to proud of themselves. And also I have another information. Beijing, you know probably China capital of China. This is north capital. Nanjing, this is south capital. So still the capital of capital may be better than Beijing or Nanjing. Anyway, it's a joke. And probably many of you Kyoto people, many of you already come to Kyoto. That is a very beautiful place. Yeah, depending on the season, you see very green season or the colored leaves season. And any season, Kyoto is very nice. And you don't be get bored to come to Kyoto. As long as many times as you come, you see something new in Kyoto. And this is the daytime, and also, so many of the temples designated as World Heritage by UNESCO. And in the evening, you also see the nice view, and also if you are lucky to see this kind of traditional kimono wearing lady. And Japanese cousin is also Kyoto. Anyway, so if you have a chance to come to Kyoto, please contact me. I'm a member of you. Thank you. And finally, I'd like to thank my colleagues. So, first, this work is supported by many of my colleagues, and I can say that just 2%, especially final part, Misao Akishiba, who is a very talented female student. She got PhD in the last year. Now she's doing postdoc in Germany. And Kentaro Sakamoto is also a PhD candidate. He do the newest peptide, and he's a little bit shy and hiding behind other people, but he's really good. And finally, I'd like to thank you for your kind attention. Thank you very much.